Hello students, welcome to my biology class. Today we will discuss on virus. Now what will you learn during this session? You will learn the history and discovery of viruses. You will learn the general characteristics of viruses, their shape and size, a brief classification of viruses, and we will study structure of a tobacco mosaic virus and structure of a bacteriophage, and end with discussion on viroids and prions. Now, there are several viral diseases, so we will have a brief discussion on some of the viral diseases also. Important characteristic features of viruses. We start with the history and discovery. These viruses, these are ultramicroscopic, infectious and intercellular parasites. They are composed of nucleoproteins. Ivanovsky, he was the first scientist to prove the existence of viruses. In his experiment, he used leaf extract of tobacco containing viruses. Now, this tobacco extract was passed through bacterial filter and then they were used or applied on healthy tobacco plants. He found that these healthy tobacco plants were also infected. This proved that it could be smaller than bacteria because it passed through bacterial filter also. Beijerinck, he coined the term virus, meaning poison, and coined the fluid as contagium vivum fluidum, which means living infectious fluid. Very important. Contagium vivum fluidum, which means living infectious fluid. What are the general characteristics of viruses? Number one, these are acellular infectious agents. They are obligate, intracellular and host specific parasites. What does it mean? They must live in any host. They live inside the host and their relationship is very specific. A virus for plant do not infect animals. So these are host specific. Viruses contain either DNA or RNA as genetic material. They can pass through bacterial filters, which proves that they are much more smaller than the bacteria. Viruses are highly resistant to antibiotics. Now the size. Smallest virus is foot and mouth disease virus, which is about 10 nanometers. Smallest plant virus is alfalfa mosaic virus, which is about 70 nanometers. Beet yellow virus is quite large, which is 1250 by 40 nanometer in size, length and breadth. Shapes of viruses. They normally exist in three basic shapes: helical, cuboidal and biennial shaped. Helical means elongated shape, as we find in tobacco mosaic virus. Cuboidal shape, they are usually short and broad, which may be polyhedral, cubical or spherical. Number third is biennial. These have both helical and cuboidal parts, as we will find in bacteriophages. Let's go into brief classification of viruses. Viruses are classified on the basis of the genetic material, or they may also be classified on the basis of their host. Let us see, on the basis of genetic material, viruses are grouped as deoxyribovira. That means these are DNA containing viruses, or ribovira. These are RNA containing viruses. We can classify them on the basis of the host also. Accordingly, they are phytophagini. That means here the plants are the hosts. For example, tobacco mosaic virus. Similarly, when animal is the host, it is called as zoophagini. For example, poliovirus. Certain viruses, they infect bacteria like T1, T2, T3, T4, etc. These are called as bacteriophage. Certain bacteria, they infect cyanobacteria and is called as cyanophage. Similarly, some viruses which infects fungi, they are called as mycophage. This is the structure of a tobacco mosaic virus, TMV. It is rod-shaped, 30 nanometer length by 50 nanometer in diameter. This structure or the covering is a capsid within which is a single-stranded RNA. This capsid is made of small units called as capsomer. These capsomer are also helically arranged around a central axis. Inside there is a central hollow axis. Now, viruses are called naked structure because this capsid is a protein covering and there is no covering over the capsid. So it is called as a naked structure. This is the structure of a bacteriophage. It looks like a tadpole with a hexagonal head and tail. It has a protein capsid which encloses a double-stranded DNA inside this. It has a cylindrical tail attached to the head with the help of a neck. Core tube is surrounded by a contractile sheath. This core tube is surrounded by a contractile sheath attached to the neck by a structure. This is called as collar. At the basal end, there is a hexagonal plate. This part is a hexagonal plate. Now, the last part with the viroids and prions. Viroids are the smallest infectious agents discovered by T.O. Diner. These viroids are actually short stretch of RNA. They do not have any capsid, protein capsid like the viruses. For example, PST viroid, this is potato spindle tuber disease causing virus. Next is prions. These are infectious protein of viruses. Viroids are short stretch of RNA and prions are infectious proteins of viruses. These prions are also disease causing. They cause Kuru disease, which is a laughing death in human. Kuru is laughing death in human, scrappy in goat and sheep, and another disease they cause is mad cow disease. So this is about viroids and prions. Now let us see some important viral diseases. There are some important human diseases, some animal diseases, and also some plant diseases. Human disease includes measles, chickenpox, polio, hepatitis, SARS, AIDS, many more. And now coronavirus disease, COVID-19, which is also caused by a virus. Animal diseases like foot and mouth disease, Ranichet, rinderpest, avian flu, and many others. Plant diseases like tobacco mosaic, bhindi yellow vein, papaya leaf curl, potato leaf curl, apple mosaic, and banana bunchy top. There are several more viral diseases. Few are collected for you. You know these terms, you know these diseases, names of the diseases is more than sufficient. Now, will you be able to fill up this table? Can you write the names of some human diseases, some animal diseases, plant diseases? You can use your previous knowledge also to fill up as much as possible. You can take your time, and also you can go back to the previous slide, learn it, and again you can write it down. Another question for you. Just now we have studied these two structures, TMV and bacteriophage. A and B of TMV, which is this part? Can you name this part? Can you name this part? And from bacteriophage A, B and C, can you name these parts? Okay, done. Thank you. Now let's check. This part is called as RNA and this part is capsid. If you have written capsomer, still it is right, because the capsid is made of small units called as capsomers. Now let us see this part, bacteriophage. A is the collar, B is the sheath or contractile sheath, and C is the end plate. So you have been successful in leveling the parts. Thank you very much. Now some viruses, example of some viruses with their genetic materials. Hepatitis B, its genetic material is DS or double-stranded DNA. Herpes virus is also a double-stranded DNA. Coliphage 5X174, it has single-stranded DNA. Reovirus has a double-stranded RNA. HIV is a single-stranded RNA. Poliovirus is a single-stranded RNA. So these are the genetic materials, single-stranded, double-stranded DNA, RNA, found in some viruses. Why I have asked you this? Very often you are asked objective questions on this, especially this part. Coliphage 5X174 having single-stranded DNA. You know DNA is normally double-stranded, but this is an exception here. Okay. Similarly, RNAs are usually single-stranded, but there is a double-stranded RNA found in reovirus. So please take care. These two examples, which will be very much important for your tests also. Now try to write down or fill up this table. Write down the genetic materials. You can do it right now. You can do it later also. Doesn't matter. Write down as much as possible within a minute. Okay, done. Thank you. You can compare with the previous slide. Now, before we leave, let us recall some important terms. Please take your pen and paper and write down the meaning of these terms. Contagium, vivum, fluidum. Next term, ribovira. You need not write the full sentence. You can write just the phrase. Bacteriophage, viroids, capsular, prions. So these few terms, write down the meaning, and now let's check. Contagium vivum fluidum means living infectious fluid. Ribovira, viruses with RNA as genetic material. Bacteriophage, virus which kills or which infects bacteria. Viroids, smallest known infectious agent made of RNA, and they are without capsids. Capsomers, these are small subunits which makes the capsid. And finally, prions, these are infectious protein particles of viruses. Now these are some questions for you which you will answer at your home. Define the term virus. Write the general characteristics of virus. How do you classify viruses? Name the 3 basic shapes of viruses. Describe in brief the structure of TMV. Draw and label the diagram of a bacteriophage. And differentiate between virions and prions. Thank you for being with me and listening. I hope you have learned about viruses. Now the next topic will be on kingdom Protista. Please visit my website. Please visit my YouTube channel. It will be available there. So thank you very much for listening to this session.